Koslicki.
Koslicki,
Yes.
So he did his PhD at Penn State University, and then did a couple short postdocs, and then started in 2012 a faculty position as assistant professor in the math department at Oregon State University. He'll be speaking on the bacterial community reconstruction via fluorescence.
Awesome, thank you. Okie, so if you guys are in the Metagenomics or Pangenomics Journal Club, then you've seen a couple of these slides before. And you might have seen a few of these slides from when I did my short little 15-minute what I do kind of talk. But basically, I wanted to sort of mix a tutorial in with a little bit of research talk, because I was like, well, hey, it's a tutorial on research, right? So it's still a tutorial. So we shall see. Basically, what I want to talk about is metagenomics. And I'm going to be talking about kind of the more traditional or ubiquitous kind of metagenomics, which is by using 16S rRNA. And then tomorrow in one of the journal clubs, I'm going to be talking about whole genome shotgun metagenomics. So if you find this stuff interesting and want to go a little bit further, then you can come to one of those. Just ask me. I think it's 2 p.m. or something. 2, 3? What we're doing? Yeah, 2. Okay, cool. So basically, what I want to do today is talk about metagenomics, talk about the typical approaches to analyzing bacterial communities, and then talk a little bit about compressed sensing and how you can use that to try to analyze metagenomics data. Okay, so what is metagenomics? So I realized previously when I've given this talk, I just dove in with the why metagenomics, which a few people here might not know what metagenomics is. So actually, let's ask people. You guys know what metagenomics is. Do you know what metagenomics is? Familiar with it? Yeah. Kind of familiar? Have you heard the word before? Yeah, okay, excellent, cool. So maybe we don't need the what, which is good because I only put a single sentence up for that, which is the study of environmental microbial communities through their sampled DNA. And there's different ways to sample the DNA and different parts of the DNA that you might be interested in. But for me, one of the most important questions to answer is why metagenomics? So the following few figures I think are pretty dang interesting. Microbes are ubiquitous and important. So let's talk about the cells in and on your body. So if you look over here, we have trillions, and these are human cells in and on your body, and that right there, that's bacterial cells vastly outweighing the number of human cells. So by some orders of measure, you are more bacteria than human. As well as there's a whole lot of coding potential in bacteria. In fact, if we look at just the bacteria that live in your gut, there are about 2 million different genes in there. And if you look at the human genome, there's about 20,000 genes. So just even the bacteria that live in your gut can do a whole lot more than what your genome can. So they've got to be pretty important. But you might imagine, why do we not walk around looking like, I don't know, very odd, cobbled together bacterial communities? The reason is that the cell size is a whole heck of a lot different. So this is a 2 scale, average human cell size, and right there you can barely see because the resolution of the monitor is an E. coli cell size. So the cells are a whole lot smaller, which is why you can fit a whole lot more of them in and on your body. A couple other reasons. Everybody loves showing this plot, and I love showing this plot, so there we go. Sequencing is cheap, so why not do it? It'll be a lot easier than culturing, especially since we can only culture a fraction of a single % of all microorganisms. So we can access identifying or access the community and try to figure out what's there by sequencing it. And metagenomics is getting a lot of attention recently. So I went to PubMed and pulled off all the articles that had titles or abstracts that mentioned just the word microbiome or metagenomics and plotted that through the years. And you can see that it's kind of booming. And so it's receiving a whole lot of attention. And you can probably see it also in popular press. You see articles in the New York Times, et cetera, the microbiome of the subway systems, or how your gut bacteria are making you depressed and all that kind of fun stuff. So anyways, a little bit more about the what. So let's go back to that. So what is it? So basically, as I said, it's studying microbial communities through their sampled DNA. They have to come from some sort of environment, and that environment could be soil samples or ocean samples, air samples, skin samples, surface samples, anything like that. It's just some sort of environment. So you take an environmental sample, and through some magic sequencing technology, you end up with a whole bunch of short reads. And from that, you want to figure out a bunch of questions. One of the very first things that you want to do is you want to ask, who's there? And at what relative abundance? Is there just one dominant phylum or is it more of an even community? Questions like this. And after you figure out this information, you can use it to do a whole bunch of different applications. For instance, like disease-associated changes in bacterial community composition. So there are many studies showing that certain communities cause a greater proclivity for different sorts of diseases, and you can cure some diseases by changing the communities. For instance, like MRSA infections. Chemical and biological threat detection. So if somebody is making some sort of biological threat or biological weapon, that actually leaves a fingerprint on the bacterial community in and around that environment. So even if they've left a few months ago, you can sample the soil and figure out what they were doing. Also, this is important for ecologists. Can you even see that? Is that below the table? Can you?
I can see that.
You can see that? We can move this table.
I can see that.
Okay. There's going to be some important formulas at the very bottom there later on, so. Okay, hope nobody wanted to sit in the front. Okay, so what we're going to focus on is this how right here. How do you go from those short reads to the composition of bacteria, which is more the bioinformatics problem. Okay, so as I said, the basic question for this paradigm is what bacteria are in the sample, how much of each bacteria, and further questions down the line. For instance, how does this change over time, over environments, over treatment, etc. So figuring out who and what is kind of like a very first step. Okay, so today we're going to talk about a couple of different mathematical approaches to the who and how much questions. Talking about questions, if there are any questions at any point, please stop and ask. Don't save them till the end. I'll be happy to answer anything. Okay, so I'm going to be focusing, as I said, on 16S-rRNA. What is 16S-rRNA? It's a certain portion of the prokaryotic ribosome. So it's involved with synthesis and translation. And so it's one particular 1,500 base pair long DNA sequence that looks exactly like this. This is an example, one of them, found in a sheep rumen. And so 1,500 kilobase pairs, but it's rRNA. So one of the most important things is its secondary structure. So it's not just a linear bunch of DNA like this. It has a secondary structure that looks like that guy. And the really important thing is if we zoom in to a few of these regions, we can see that there's these really interesting hairpin turn structures here. Now, why is that important? Well, since this is involved with such a basic process, protein synthesis and translation, it turns out that all the bacteria that we know about has 16S-rRNA. So it's ubiquitous, and every organism has it. But these little hairpin structures have a very unique property to them. That unique property is the following. So let's take a whole bunch of these 16S-rRNA genes, and let's align them all to the E. coli 16S-rRNA. So I took these and I'm going to align them. What you get is this. So this is position relative to the E. coli genome. And over here is some unimportant measure of variability. It's like reverse entropy or something like that. Details aren't important here. What you should notice is that higher peaks means more variability in that particular region. Lower peaks means less variability. So you can see after you've stacked up a whole bunch of these different 16S-rRNAs, there are some regions that are very highly conserved across the various organisms. And then there are regions just flanking to it that the variability is really high. So the whole sort of point is, well, since every bacteria that we know of has a 16S-rRNA, we can design a primer to latch onto this region because it's pretty well conserved. So it looks roughly the same. And then sequence into this variable region. And then since that's pretty variable, we can use that to distinguish and identify which organism is which. Okay, this is accomplished via amplicon sequencing. So you design a primer, as I said, just a short piece of DNA. It looks like this. This is not an actual primer, but a piece of an actual primer that binds just adjacent to a variable region. So this is that same picture. Let's go ahead and concentrate on one of the variable regions. We zoom in here. What the primer does, it's a reverse complement to one of those highly conserved regions here in black. It sits on here, and then you use amplicon sequencing to sequence into this variable region, and you'll end up with a read. If you're using 454 technology, something about 450 base pairs long, and the reads will look like this. So basically, the whole paradigm is you take your sample, you throw it on one of these sequencers, bind the primer. Yeah.
Could you go back to the 450 for?
Absolutely.
Because actually the example, this loop is definitely not as long as 450 base pairs.
Absolutely. Yep. Yeah. Good observation. So if we step back to this guy, you can take advantage of the fact because there are multiple variable regions. What you can do is you can design a primer to latch on right here and then sequence multiple ones. Yeah, thereby actually increasing your distinguishing power. Yep. So typical studies will target actually a number of these different variable regions, if possible. Good observation. Yeah.
So the preserved region, like is there any mechanism or evolutionary sensitive structure, like why those are conserved and the other ones are like variable?
Being a mathematician and not a biologist, the only thing that I can guess is that these regions of the secondary structure are very important to keep this, conserved sequence identity to be able to have this sort of structure. In this region, it's not so important, because you can have a little bubble here or something that's not going to really change its mechanism. But that's just a wild guess on my part. Yep.
So that region is more susceptible to like
Accumulation of errors, yeah.
or like non-genomic mutations?
I would assume. Yep. Yeah, so what I'm skipping over here is a whole lot of other important biological details. For instance, like copy number. So you can have different numbers of copies of 16S rRNA. You can actually have different copies that have different nucleotide compositions of 16S rRNA. So a bunch of little details, but if you're interested, you can ask me at some point. Okay, so there we go. We have all our bacterial DNA sitting around. We hit it with a primer. We sequenced along into the variable region, and we get out a whole bunch of sequences that look like this. So since we used a primer, the first part of all the sequences are basically the same. All right, so all the blue is basically the same. That was the primer. And then we sequenced along, and these regions are different because those are the variable regions. And the whole point is to now try to figure out, okay, this is supposed to serve as a barcode or a fingerprint for the organism. How are we going to distinguish it based just on these ACs, T's, and G's here? And yes, I know I did not put 450 right here, but that would kind of be long on the screen. Okay, well, what about known information? Let's talk about database sequences. So, Ribosomal Database Project is a project, I think at Michigan State University? Some university starts with an M, which is collecting a big old database of these 16S rRNA. So this number is a bit old. It's like 1.5 years old, so it probably has a lot more than 2.6 million 16S rRNA sequences. But basically, there's a lot. You sequence it, you find a new one, you annotate it, throw it into the database. It's about 30 gigs when you download the whole thing, because remember, they're relatively short, 1.5 kilobase pairs each. But unfortunately, it's kind of a dirty database. What do I mean by that? Well, even though they try to do a really good job of curating this, there are a whole bunch of dubious entries. For instance, this is something that probably is an actual 16S rRNA for this particular organism. It's roughly the same length. They could identify which organism it belongs to. If you look at the metadata, it's been error checked, et cetera, et cetera. And then you have entries that look like this. 270 base pair sequence. They don't know what organism it belongs to. They didn't do any error checking or anything like that. This is just the raw sequence from some sort of Yellowstone geothermal vent sample. So,
You don't have any QC before you really upload the sequence to the database.
I don't know what QC they have. They say that they try, so I believe them. But things do sneak through. Yep. This could have been before it was really implemented well, because this was from, way back when. Okay, so we're going to try to leverage this information, this large database of previously known 16S rRNA sequences, to answer the question of which bacteria are in my sample. Okay, Well, we are flying through, which is fine. Typical approaches. What do people usually do? Well, the Bayesian approach is one of the most popular right now. Bayesian approaches are implemented in common computational software packages to analyze bacterial communities like CHIME and Mother. Bayes' theorem basically goes through and, or the Bayesian approach goes through and classifies each sequence read by read. So the one I'm going to be talking about here is the Ribosomal Database Project's naive Bayesian classifier, which is a massive mouthful to say, especially 5 times fast. So instead, I'm going to say RDPs and BC. So RDPs and BC, that's that guy. Okay, so the whole paradigm is like this. Here's your input sample of sequences. You want to identify which organisms these guys belong to. You want to figure out who's in my sample and what relative abundance. So you go, you take off your first read right here, and you hit it with Bayes' theorem. So it's a Kamer-based technique. I don't want to go through a ton of the details, just give you kind of an idea of what's happening here. So this right here is a Kamer. It's part of this read right here. And you want to find the probability that that Kamer is found, given that you're looking at genome I, and then maximize this probability over which genome you're classifying to. So this maximization right here is over a very carefully designed database of known sequences. So RDPs and BC uses a database of about, yeah, you can't read that, 10,000 sequences. So right there where you can't read it, this I right here ranges over 10,000 sequences. So you can classify this one sequence to 10,000 different possibilities, or 10,046, based on maximizing probabilities of Kamer counts matching. You do that, and it says, oh, most likely it belongs to, you do this bunch of math, it most likely belongs to this phylum, this class, all the way down to that genus. Usually, genus level classifications are still dubious. You can't really classify to the species, and definitely not to the strain level, just using 16S rRNA. So genus is about like the best you can get. Okay, so naive Bayesian classifier told us this classification was the one that maximized this probability. Great, done with one sequence. Go on to the next one. Do that bunch of math again, get another classification. Go to the next read, do the classification, et cetera, et cetera, et cetera. After this point, you take all those classifications, so this is the classification of the first read, classification of the second read, classification of the third read, and then you basically just tally these up and divide by the total. So for instance, at the phylum level, you count all the different unique phyla and then count how many times they appear in your bunch of classifications, and you can get that little pie chart of who's there and how much. So you take these guys and you end up with a taxonomic profile. And so this is what a taxonomic profile looks like. It's a list of different taxonomic ranks and tax IDs, different taxa names, as well as their relative abundance. So for instance, this taxonomic profile says that 98.2% is bacteria, and let's see here, we have mostly proteobacteria and a bunch of firmicutes as well, etc. So when you're talking about taxonomic profiling, this is the end result. The way that the Bayesian classifiers go about it is they first do a read by read binning. So bin, like you stick it in different taxonomic bins. Take a sequence, stick it in a bin. So it does a read by read binning and then summarizes that information into a taxonomic profile. That make sense? Questions about that guy? No work? Yeah. Mm-hmm.
It means the percentage, the sum is not 1, right?
Great observation. Yes, it is not 1 because some of these sequences, you don't know which one they belong to. So for instance, when you try to maximize that probability, maybe the maximum probability is something like 10%. And you have some sort of threshold saying, well, if it's below 80% confidence or something, I'm going to say I don't know what it is. Yep. So exactly. Some of them you don't know.
Is it because of the error in the read, or it's like unknown, it's possible that database doesn't have that particular?
It is extremely likely that the database does not have the exact sequence that you've seen. Yep, because remember, we're only classifying to 10,000 different sequences. And back like 3 years ago, we had on hand 2.6 million different ones. So it's actually pretty likely that it's not going to exactly match. But you hope that it at least matches at the genus level or above. Yep. Did I see another question somewhere over there? Nope. Okay. Cool. So a few different criticisms about this. First off, it's somewhat slow, but easily parallelizable, because you got to go read by read by read by read, as well as the fact that it's incorporating very little known information. You go and you classify this sequence, and then you forget everything that you've already done, go to the next sequence, and in isolation, classify that guy. And then forget everything you've already known and go and classify the next guy, et cetera. So you're really treating each read as completely independent from the other ones, which it's not, because it comes from the same sample. So for instance, instead of a naive Bayesian classifier, what if we could get some sort of prior on the distribution of the taxonomic classification, use information from other reads, et cetera. As well as the fact that, as I've been saying, RDP's database, you can barely see it down there, has 2.6 million sequences, and the training set is only 10,000. So we know of 2.6 million. Actually, now I'm pretty sure this is like 5 million. But anyways, we can still only classify to 10,000 sequences. The reason why this is because for each read, we have to go and look at that probability over each one of the genomes, right? So if you try to make your training database huge, like 2.6 million, the complexity of that computation is going to be big O of the number of reads times the number of training database sequences. Yeah?
Can you perhaps do some iterations?
Yeah. Yeah, you...
First, find like, know that you're in the right city, then in the right neighborhood, then in the right house.
Yes, yes, yeah, you could absolutely do that. One immediate thing that might happen is that usually these samples are pretty diverse. So unless you're looking at some very niche little community, the right neighborhood is actually pretty big. But yes, that is definitely one improvement that you could do. I haven't seen that. Oddly enough, naive Bayesian classifier, just this plain vanilla approach that I'm mentioning here, seems to work well enough that people aren't working too much on trying to improve it. That doesn't, yeah.
Very naive questions.
Please.
What's the difference between this? How do they select the Pensel sequences from the 2.6 million?
Yes, great question. So they're selected for different, to optimize different criteria. So for instance, over the tree of life, they want to try to do some sort of uniform sampling over different bacteria so that one particular, clade is not overrepresented, as well as they don't want to pick sequences that are too closely related to each other, because then that's just going to confound your classifier, basically. Yep. And then they also want to minimize the number of sequences that you're using to be able to speed this method. Okay, so as people are observing, this is probably not the best that we can do, so let's see how we can try to improve this. Okay, so what do we want to do now, halfway through? Let's talk about compressive sensing. I mentioned this extremely briefly during my 15-minute talk a couple weeks ago. What is compressive sensing? So take off our biologist hat and we're going to put on our mathematician hat, and we're going to talk a little bit of linear algebra. So everybody here is taking linear algebra, right? Grad students take linear algebra, right? Yes? No?
It's a long time ago.
It's been a long time ago. Okay, let's try and refresh our memories. We have a matrix, A. We have a vector, X. We have another vector, Y, right here. And we can talk about this later. This is basically the situation that I'm going to be looking at. So our matrix is this short, fat guy. Our X is this really long one, and our Y is this very short one right here. Now in math, what do you always solve for? You solve for x, right? X is our unknown, the thing that we're trying to solve here. A is known, y is sampled. So we use some known information and some sampled information to try to figure out what this x is right here. So we know all these, we know all those, and then we end up with a big system of linear equations, and we want to solve what these variables should be to satisfy that equation. However, if we're in this situation right here, which is referred to as underdetermined, we can have infinitely many solutions, because think about it. Your sampled data, like over here, we have 4 known points, and we're trying to solve for a bunch of variables. So we have way too many variables for data points that we have. So you can imagine that this is horribly underdetermined, and you can have infinitely many solutions. Don't give up. Because if you make one little assumption, and this is like the basis underneath compressed sensing, you assume that x has many zeros. Whenever this vector right here has a lot of zeros, we refer to that as a sparse vector. So sparse, lots of zeros. Then you can actually solve this equation ax equals y uniquely by not asking for any vector, but asking for the sparsest vector to solve ax equals y. So try and satisfy ax equals y, that's sparse as possible. Everybody still tracking? Good? Okay, cool. And we're going to try and do that via some sort of convex optimization. It's like, oh, sweet, I can write down an optimization procedure. Let's minimize the number of non-zero entries in x subject to satisfying the equation ax equals y. Right? That sounds like a good optimization procedure. Minimize the number of non-zero entries subject to satisfying ax equals y. Unfortunately, that's NP hard. So there's no known polynomial time algorithm to solve this thing, and on top of that, if we did find one, we could solve any other NP hard problem.
By the way,
Yes?
if you define the next that minimizes this expression, you know it's unique?
So in a minute, we will modify this, and then yes, it will be unique. Yep, right here, as I've written it, no. Yep. So geometrically, if you like thinking in pictures, what's going on here is you have a hyperplane, so a plane defined by this equation ax equals y, and minimizing the number of non-zero entries is basically asking that you get as few intersections with coordinate axes as possible. And you can imagine that to do that, you just kind of have to try all different configurations until you find one that's the optimal, and that's why it's very difficult. However, so Emmanuel Condes, Donahoe, and Terry Tao, which is the super famous mathematician who's just sitting over there, which is kind of really cool, on the same campus as him. But anyways, came up with this great observation that we can just replace this L0 norm with a proxy called the L1 norm. So all we had to do is switch this little 0 to a 1. This norm right here is the sum of the absolute values of the entries in X. And you might think, what in the world does that have to do with minimizing the number of non-zero entries? Well, it turns out that mathematically you can prove that as long as this matrix A right here satisfies some nice geometric conditions, the most traditional being something called the RIP, or restricted isometry principle, meaning that as a linear transformation, this A right here does not deform space too much, then you can show that this optimization procedure right here, called basis pursuit, is convex. There's a unique solution, and you can get in polynomial time. So which is really cool that you can go from NP hard, just switch your norm, to, hey, look at this, in polynomial time unique solution, which is pretty awesome. And the idea here is that we have this hyperplane. Instead of intersecting coordinate axes, we want to intersect this prism guy right here. And so you can imagine that you can use a convex optimization procedure. You just sort of walk along some sort of hill climbing technique if you wanted to. The optimization procedure that I'm going to be talking about is this other one, though, called basis pursuit denoising. So what did we do here? Well, in the previous step, we said subject to ax exactly equaling y. But given that there's always measurement error and some noise going on, we don't want ax to exactly equal y, we just want it to be close. So that's what this optimization procedure right here, it's called a regularized optimization procedure, because I still have this L1 norm here, and that's what made things work. And then I say some constant times, and you can think about this L2 norm squared as just the distance between AX and Y. So we're trying to minimize this quantity. So if this lambda right here was huge, like 10 billion, then we would want this thing to be really small. We would want AX to be really close to Y to obtain a minimum. So this optimization procedure right here is robust. So if we did AX equals Y plus some noise, some epsilon error, it would still work. And you can imagine, as lambda approaches infinity, this basis pursuit denoising approaches basis pursuit in the previous slide. Why would you want to do this? Why can't you just use least squares? I mean, come on, it seems to work well. Well, this sort of basis pursuit denoising has much superior performance, much better performance on sparse signals. So here is going to be our sparse signal. So this is not mine. Whoops, I stole it from the survey paper. But basically, this is the thing we're trying to reconstruct. So you can see that most everything is 0, right? But it has a few non-zero entries. And then what you can't see here is the measurement. This is our x, the thing we're trying to reconstruct. You can't see the AX here. But basically, there we go. If you use the basis pursuit denoising, where your data is Y, your measurements are this AX right here, and your X is your fit or reconstruction, you get something that looks like this. So notice the blue was the original signal. The red is our reconstruction. And look how nicely the red overlaps the blue, right? It's like a perfect reconstruction. We solve for X pretty much exactly. Well, what if we did least squares? So this is least squares, right? Minimize that quantity. That's what it looks like. It's just complete garbage, right? Because we're terribly overfitting the data. So for sparse signals, basis pursuit denoising and these other sort of compressive sensing ideas are a whole lot better than doing least squares. But is it? There'll be a fun little caveat later on. And you might be asking, yeah.
Well, this is like a nice setting where you know what sort of vector you need to recover.
Mm-hmm.
But in real life, how do you get the accuracy of the...
Great question. So it turns out that the next part that we're going to do, which is applying this stuff to metagenomics, that assumption of reconstructing a sparse vector is actually almost trivially satisfied. You have a database of 2.6 million sequences. You take some sample. Of course, only a small fraction of that whole 2.6 million is going to be represented in that sample. So it's a pretty easy assumption saying that it's a sparse vector. And then that matrix A that we talk about, we can design it to at least partially satisfy some of the properties that are theoretically needed to prove reconstruction. Yep, so yes, there is hope, in other words. Okay, so you might be asking, what in the world does this have to do with metagenomics? Let's try to make that connection right now. So we're talking about AX equals Y. Basically, what you want to think about is this A, I said was a previously known information. We're going to try and stick our 16S rRNA database right into there. This X is a concentration of bacteria, and this is exactly what I was saying, that it's a sparse vector. That's the thing we're trying to solve for. And so since this massive database was done from thousands of samples all over the world, you can imagine that if you take one little skin sample, of course, you're not going to get the same sort of microbes that you would in a Yellowstone hydrothermal vent. So this vector is going to be sparse. And then we're going to try and fit our metagenomic sample into this Y right here, and then simply hit it with some sort of compressive sensing technique and solve for AX equals Y. The way that we're going to do this is using a Kamer-based approach. So we're going to fix the Kamer size. So AC's, T's, and G's of length about 6 here. And then I'm going to tell you how I'm going to construct this A right here. So you go to your ribosomal database project of 16S RNA sequences. You take one of them that belongs to known bacteria number 1, and you calculate the frequency of all the Kamers in it. So you calculate frequency of AA, all the way down to TTT, TTT, and you get a probability vector, and you stick that on to A. Then you go to your next known database sequence, stick it on, et cetera, et cetera, et cetera, and you end up with this matrix. So first off, ooh, we could do a quiz for who's paying attention. How many rows does this have? 4 to the power 6, exactly. Roughly how many columns is this going to have? How many known bacteria do I know of? There's 16S RNA sequences. Yeah, hundreds of thousands. We could use the training database that RDP does, which is like 10,000, or we could use all 2.6 million of them, right? So this can range from 10,000 to 2.6 million different columns right here. So we're definitely in that situation of a matrix that's very short and wide.
Did you spin out the columns based on entries in the database being very similar? Like, the same bacteria reporting multiplying the data frame? And if not, does that cause problems with the coherence properties of the one you use it for?
Yes, absolutely. So the interesting thing is, theoretically, yes, it causes problems. Practically, nope, it doesn't. And the reason why is that if you have two bacteria that are very closely related by their 16S RNA sequences, then with extremely high probability, they're closely related in their taxonomic composition as well. So if you have some non-uniqueness, like you could put some weight on this organism or on that organism, it's not going to actually change anything downstream because they're both exactly the same organisms down to the genus level. Yep. Yeah, typically you don't try to classify below genus level using 16S RNA. Yep. So it's kind of like that saves us. Yep. Okay, one thing to observe about this matrix right here is that the column sums are 1, right? Because these are probability vectors. Then we can construct our Y right here by giving the sample. You just calculate the frequency of all the k-mers in it. So you have the sample of all the reads, and you compress it down by calculating frequency of all those k-mers. This is the compression part of compressed sensing. We've reduced our whole sample down to a signature that just consists of frequencies of 6 mers. And then you can show that AX equals Y. Just calculate it out. So we are exactly in this compressive sensing regime. Great. We can do this. We can now use basis pursuit denoising. Oh, unfortunately, it's really slow. So all the algorithms that they had up until now to use basis pursuit denoising, using a matrix with 4 to the 6 rows and 2.6 million columns, agonizingly slow. But the cool thing is, remember how earlier we changed the L0 norm to the L1 and suddenly something that was really hard became easier? Something analogous happens right here. What happens if instead of the L1 norm, we just stick a square right on top? Then, due to the properties of the vector that we're trying to reconstruct being non-negative, and the sums of the columns of this A being 1, or bounded between a constant, you can take this and reduce it to this. So this is exactly mathematically equivalent to that. This right here is a matrix where the first row is all 1s. And then we take square root of lambda on A and stick it right below it. This right here is a column vector. The very first entry is 0, and then we have Y underneath it like that. What does this look like? AX minus Y. This is least squares. What? You can take basis pursuit denoising. As long as you have these assumptions, you can solve it using least squares. Which is kind of really cool, because this is an observation that hadn't been made before, and is one of the reasons why I like doing mathematical biology, because usually you think about the math always helping out the biologist, creating new algorithms, things along those lines. And here's a cool little example of the biology actually informing the math. Nobody had observed before that you can reduce a compressive sensing regime down to least squares. So, whole point about this guy, there's a reference if you want to look at it, sparse reconstruction can be performed by modified least squares. So, beforehand we saw this picture where he said least squares is bad. But if you just tweak it ever so slightly, stick on this row of 1s and a 0 right there, boom, all of a sudden it becomes a whole lot better. And the extra cool thing is least squares is a super optimized algorithm, and it can handle 4 to the 6 rows in hundreds of thousands or a million columns.
How do you choose lambda?
How do we choose lambda? For 16S, it's actually not sensitive really to the lambda choice that you do as long as it's above 100, interestingly enough. For other sorts of data, if you try to modify this regime to whole genome shotgun sequences, then you actually have to use an extra training procedure on top to choose the optimal lambda. But for 16S, it's pretty insensitive.
But it thinks the key transformations in this list, you change the norm 1x
Yep.
to a square, right?
Yes, yes, exactly. And in this paper, we show that by doing that, you don't change the answer at the end. The answer that you get is the same answer that you would get if you did not have the 2 there. Yep.
So this modified least squares thing.
Mm-hmm.
In the things you're minimizing, you have standard least squares and this other term now squared. Is this ridge regression?
Is this what?
Ridge regression?
It's related in form, yeah. Yeah, all these sort of regularized regression techniques are very similar to each other. Just depends on exactly how you square, don't square, which norms you choose, et cetera.
But the one norm of x squared,
Mhm.
then x is non-negative.
Mm-hmm.
The same as just squaring x. So where is it different than ridge regression?
I would have to look up ridge regression again just to see. I'd have to compare the formulas.
Okay.
Yeah, it might indeed be exactly the same,
Okay.
but I don't know. Okay, so we wrap this all up into an algorithm called Quicker. Rows, k-mers, columns, known bacterial 16S RNA sequences. Entries here are the frequencies. That's what we're trying to reconstruct. That's the information that we're given. We use this optimization procedure. And it took a few weeks to come up with the name Quicker, which stands for a quadratic iterative k-mer-based reconstruction technique. Because quadratic, iterative, because the way you solve this is using an iterative method, and it is a k-mer-based bacterial community reconstruction technique.
And you started with the name then.
Actually, no. I sat down and I wrote all the different adjectives that could possibly describe this method, and then I took their first letters and did one of those like New York Times puzzles or whatever and tried to just arrange them into something while also looking at a list of adjectives for fast. Exactly, yep. So, because the whole point is this is ridiculously fast. How fast? Well, let's look at it. It has sublinear execution time. So here is a log plot. Over here is a number of reads in the sample. Here is the number of seconds to classify. So RDP's NBC is linear right here. Quicker is pretty dang quick. How quick? This right here is a sample of over 1 million reads. So over 1 million reads classified in under a minute. Whereas using RDP's NBC, that would take about 10 to the 4th seconds. So it's pretty dang ridiculously fast. Yeah, we'll look at that next. Great lead into the next slide. Let's look at it on simulated data because that's where we know the ground truth. I'll take a whole bunch of simulated data sets with a bunch of different reads, a variety of different simulation parameters, about roughly 800,000 reads per data set, which is not unreasonable for 16S RNA. Now remember, this AX equals Y, we get to create that A ourselves, right? We get to choose which sequences we pick to make the columns. So if you used the same sequences as RDP's NBC, so the same 10,046 sequences, then what do we get? We get this. Okay, let's look at this plot right here. On the y-axis is L1 error. So if X is the ground truth, X star is our reconstruction, and we just take the L1 norm of the distance of those two guys and plot that versus taxonomic rank. So lower is better. This is an error. Lower is better. Blue is quicker. Red is RDP's NBC. So for lower taxonomic ranks, quicker is doing better. And at the family level, they're about identical. In the genus level, quicker is doing worse. But remember, the whole point was that this algorithm is very fast. And we can use very large training databases. So let's instead use all well-labeled sequences in RDP's database. What does well-labeled mean? Well, I went through and used somebody else's tool to be able to find all those 16S RNA sequences that are annotated down to the genus level. I just took all of them, all 275,000 of them, and trained this A matrix right here.
If you do that,
Yeah.
I think you will get several columns in the A matrix that come from the same sequence.
Yes. Yes, you will. And
Kind of metal with your sparsity.
yes, it meddles with your sparsity. It also meddles with the uniqueness part, right? Thankfully, though, since at the end of the day, we just want to summarize that information. If we have two different species, it's the same genus. So I don't care which one I choose. And it's also block sparse, right? You can put all those organisms that are the same species underneath the genus. And then just think about your matrix as just a bunch of genera, and that's sparse. So, but anyways, apples to apples comparison, apple to oranges comparison right here. Notice every taxonomic rank quicker has less error. And the reason is that we can use 275,000 training organisms, whereas this red line right here is only using 10,000. By the way, if you tried to retrain RDP's NBC with all 275,000 well-labeled 16S RNA sequences, then it would take years per sample to classify. And so it would take about 216 years to finish this experiment right here. So I didn't do that. But the whole point is, when you have a faster algorithm, you can use more training sequences, and that helps. Excellent. We have a couple more minutes to talk about a couple different improvements that you can do. First off, K-mer frequencies that we use to create this database A right here. We were just using the frequency in the whole 16S RNA sequence. But these K-mer frequencies are not stationary as you move along the sequence. So this is a 16S RNA sequence. And let's take sliding windows like that. And we can look at the 6-mer frequencies in the first, or 6-mer would be very hard to visualize, so I'll just do 1-mer frequencies. In the first window, and in the second window, and third window, fourth window, etc. So on the x-axis is position along the sequence. This right here is a bar chart of the relative abundance of these K-mers right here. And you can see along the entire sequence, it fluctuates, right? And that's just for 1-mers. When you move up to 2-mers, they fluctuate a whole heck of a lot, which makes total sense because, you know, some regions are variable regions. And so that's more like a Bernoulli process with accumulating errors. So the frequencies are going to change as opposed to the conserved regions, et cetera. So it makes total and complete sense. So we can actually extend this matrix A to incorporate that information in the following way. So instead of just putting the K-mer counts of the entire sequence, you can do the K-mer counts of the first window, K-mer counts of the second window, et cetera, and lump all those into known bacterial sequence number 1. And then do that for the second one, et cetera, et cetera. So here again, you have to take advantage of block sparsity, right? Because, of course, these individual columns are going to be similar to each other because you're using a sliding window. However, you can partition this matrix into blocks. And the whole point is, because of that, you get a decrease in error, right? So this is the original quicker one. This is when you're using sliding windows, and you get a decrease in error. And lastly, a little bit of machine learning pre-processing always helps. So let's go ahead and pre-cluster the reads. So you take all the reads and use a really simple k-means clustering. It's relatively fast. And then you end up with a bunch of clusters. You use quicker or sec on each one of those clusters. You recombine them like that. And what happens? You get an even further decrease in error. So plain vanilla algorithm, use sliding windows, pre-cluster the reads, gets better, 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 better. So, conclusion, new math leads to a new paradigm in bacterial community reconstruction, which is why I like being a mathematical biologist, because you can take advantage of this kind of new stuff to lead to new algorithms, which is always fun. These sort of compressive sensing techniques are sort of like a big data solution in that they're sublinear in the computational complexity. You can classify the whole sample instead of read by read, because remember, we're compressing everything down into K-mer counts. So we're not looking at individual reads. The only bad thing is trying to get like p-values or confidence bounds on these things are nastily difficult. So there are theoretical results that have to do with confidence bounds on least squares, but they're really difficult to implement. More info, you can go look at all the papers, stuff like that. If you're interested in actually playing with the software, it's written in Julia and Python and in Docker at GitHub, and I have a Galaxy running as well. So, whoop, very quick things. There we go. Yeah, we're not going to go all the way through that again. So I'll end and say thank you. You guys had a great amount of questions when we were going through it, so any questions?